Okay. So we were talking about the examples of organisms which undergo fission. Few examples I have mentioned like amoeba, euglena, paramecium, leishmania. These are few organisms which undergo fission. Now we will talk about the types of fission. What are the different types of fission? According to the number of organisms produced, we divide fission into two main categories. One is binary, another one is multiple fission. So in binary, as the name indicates, two daughter cells are produced. And remember that, बच्चे, in case of fission, the parent organism vanishes and new organisms are formed. Okay. In case of multiple fission, more than two daughter cells are formed. Now we will see the basic procedure by which this process of fission occurs. Method of fission. Method of the process of fission. It takes place in two steps. Number one, during the process of fission, the first step which takes place is division of nucleus. Division of nucleus, which is present in all the eukaryotic cells. Second step of fission is division of cytoplasm. So in the process of fission, first the nucleus will divide and break up into two or more nuclei, and then after that the division of cytoplasm is followed. How does this happen? We will see with the help of examples. First example which I'm going to take is of amoeba. The thing which you need to remember about fission in amoeba is that because we know the body of amoeba is irregular and it is a unicellular organism. It has a very basic body design with single celled body, cytoplasm in it and nucleus in it. Okay. So the fission process in amoeba can take place in any plane. Fission in amoeba can take place in any plane. What do you mean by that? It means that the body is irregular. The fission can start from here also. It can take place from here also, from here also, okay, from here also. This is called as the fission taking place in any plane. Okay. There's no specific spot for the fission to start. Now, what are the various steps of fission in amoeba? First step should be the division of nucleus. Before the nucleus divides, there is a deep invagination which occurs in the nucleus. This is called as deep curve or invagination. After this, the nucleus will divide into two nuclei. And then finally, and after the division of two nucleus into two nuclei, there will be a division of cytoplasm also. Okay. And finally, the two daughter amoebae will be formed with one nuclei in each and cytoplasm too. So this is a process of fission in amoeba. These diagrams are very important because usually students, they take it lightly and don't go through them when they are studying for their exams. But you should know that the division of nucleus occurs first and then the division of cytoplasm occurs during fission, right? Now, second case of fission is leishmania. Leishmania is an organism which is single-celled but with more structures on its body as compared to amoeba. We don't get to see any external structure in case of amoeba, okay? But we get to see these structures in leishmania. So what do we say that? The body structure of leishmania, though it is also unicellular, is little more complicated than amoeba. How does it look like? It looks like this, okay? And then a whip-like structure on its body with the nucleus inside. This whip-like structure is called as flagellum, which was not there in amoeba. Now, what two things which you need to remember about leishmania? Number one, because the body is more organized in case of leishmania because of the presence of this flagellum. So fission takes place at a specific spot. From a specific spot. Right? And second thing which you need to remember, this is very, very important, and this comes in your exam. We know that amoeba is a protozoan which causes dysentery, loose motions. What is caused by leishmania? You should know the disease which is caused by leishmania. The name of the disease, it causes Kala Azar. So what sort of question can be asked from this part? Name the organism which causes Kala Azar, or leishmania causes which disease? So if you know the disease which is caused by leishmania, you will be able to answer it, right? Now, how does the fission process start in leishmania? We know that it has a more complicated body because of the presence of this flagellum. So obviously we cannot cut it like this or in any plane. Otherwise, while dividing, where will this flagellum go? So we'll have to take care when we show the process of fission in leishmania. It starts from flagellum. So this is the parent leishmania. First, the flagellum will grow. Nucleus will show a vegetation. Then the body of the leishmania will start splitting into two flagellums, two nuclei, and this vegetation becomes deep. And hence, how will you show the deep vegetation? So, anyhow, here are two nuclei, and finally, it will split into two daughter leishmanias with One flagellum, it is looking like a carrot. And definitely there is cytoplasm present. So first the cleavage of nucleus and then the cleavage of cytoplasm takes place. So that is how the process of fission takes place in an organism which is not having a simple body design like amoeba. Have you understood this process now? Any doubt in fission? So this was the first method of asexual reproduction, which is adopted by unicellular organisms. In unicellular organisms also, there are two cases. The one in which the body is very simple, like in case of amoeba, and the one which has little more complicated body design. But both of them they are unicellular organisms. Now the second type of fission is multiple fission. We know that in case of multiple fission, the body of the organism splits into more than two daughter nuclei. Okay? So in which organism does it occur? Example we will be doing of the one which is given in your book. And there are few more examples which you can write of multiple fission that I will write on the screen. The first example which is given in your body in your book is Plasmodium. Plasmodium vivax, which causes malaria. We know that, and because multiple fission has to take place, so the body structure of Plasmodium will be even more complicated than Leishmania and amoeba. So the body of the Plasmodium is made up of different types of cell. It looks like this if you draw it. Different layers of different kinds of cells and the nucleus in the center. Now when Plasmodium has to divide, this first we know in both the cases, whether it is multiple fission or it is binary fission, the first step would be the division of the nuclei. So first the nuclei, but here the difference will be that this nucleus will divide not into two nuclei, but more than two nuclei. Lots of nuclei will be produced in the body of the organism. And then because we know that cytoplasm is also present, what will happen to the cytoplasm? Cytoplasm will divide and surround each nuclei. Right? Like this. And finally, when the organism will undergo multiple fission, these nuclei will come out along with the cytoplasm and membrane around it, forming number of daughter cells. So that is how the multiple fission takes place in Plasmodium. Now, what are few more examples which you can write of the organisms other than the Plasmodium? Yes, Niket?
Ma'am, is there any unicellular organism which is visible through eye?
Which is visible through?
Through naked eye.
Yes, there are. There are. But before that,
ma'am, these three are not visible, or are they visible?
With the naked eye? No, you have to see it under a microscope.
Okay.
All of them, they are microorganisms. So examples are amoeba. Amoeba can also undergo multiple fission, but under special conditions. Okay? Under special conditions. Otherwise, the usual method of or the normal method of division of amoeba, reproduction of amoeba is binary fission. But under special conditions, amoeba can also reproduce by multiple fission. And then next example is Chlamydomonas. Chlamydomonas is again a organism which looks like this, and it also has a flagellum on its body. Okay, so here again the complication in the body can be seen. So obviously the multiple fission will occur at a definite pain. Other than that, there are certain platforms, certain species of alga which undergo, some alga also undergo multiple fission. Okay, so these are few examples. Now binary fission में भी बच्चे आपकी book के अंदर only two examples are given. One is of leishmania and another one is of amoeba. If you want to write more, please do write. I'm going to add few more examples. You can note it down. If we are talking about planaria. Planaria also you can add. Yes, Niket. Niket, you want to ask something? Niket, do you want to ask something? So this is all about fission. Now we will start with the second method of asexual reproduction. That is fragmentation. Similarly, just like fission, we will start with the definition of fragmentation. But here definitions, they are usually asked in the exam. So we'll have to know the definition of each type of fission. Okay. Niket, you want to ask something?
No, ma'am. It is taking time to lower down.
Not a problem. So definition of fragmentation will be, in this process, the parent body breaks into or splits into two or more fragments. Now what is the reason? अब automatically अपने आप तो fragmentation होगी नहीं, ठीक है? There would be some cause of these fragments to break. So the cause would be due to the wave action, because fragmentation usually occurs in algae forms, filamentous algae. Algae are of different types and they are multicellular in nature. Okay, remember that fragmentation occurs in multicellular organisms. In case of alga, like Spirogyra, this fragmentation occurs due to wave action. A strong wave comes and break the parent organism, or due to the death and decay of any fragment in the body of the organism can cause the process of fragmentation due to either wave action, or death and decay of older parts. Now, various examples which we can add of the organisms which reproduce by fragmentation are, you can add sponges, we can add flatworms, and some algae. A very good example which we can use to explain this process is alga spirogyra. But before we start with the process of fragmentation in spirogyra, you should know the structural body structure of a spirogyra. And it also comes to label in the exams. So I'm going to draw the structure of a spirogyra first, and then I will label it. Okay. A process you have already understood. This is the structure of a spirogyra with, this is mucilage. Mucilage is a sticky layer which is present in the algal form. That is why they are little slimy in nature. And this is one cell, this is another cell. Inside it, there is another layer which is called as cell wall. This is the cell wall of spirogyra. Spirogyra is an algae. And this is one fragment, this is another fragment. Fragment means pieces. That is why we say spirogyra, it is a multicellular organism with basic body design. That means the body is made up of number of cells, but the tissues are not formed. This is cell wall. What will be there? Because it's a plant, alga is a plant, multicellular plant organism. Inside this, there will be another layer which is called as cell membrane. So I'm going to just label it first. This is mucilage. The inner layer is cell wall. Inner than that, cell membrane or plasma membrane. Cytoplasm is not scattered in the UN of this. Rather, how is cytoplasm present? So these are the spaces which are present. These are the vacuoles. Okay. And similarly here, large vacuoles are found because it's a plant cell. And now on this, there are cytoplasmic strands. Now cytoplasmic strands, they contain the cytoplasm inside it. Now cytoplasm will be present here in these strands. Here also. Similarly here. Inside these cytoplasmic strands, there are pyrenoids, तो चलो आपको नहीं आएगा. The nucleus is present in the center of the cell here. Okay, here. So now I'm going to label the further parts. This is called as vacuole. This orange structure, this one, is called as cytoplasmic strand. बच्चे ये आपकी बुक के अंदर नहीं है, but it is there in your practical file, and it usually comes to label. And this one is called as, this one is cytoplasm. So cytoplasm is not scattered here and there. It is well organized in these strands. And this, last but not the least, is the nucleus. Other than that, in these cytoplasmic strands, there are special structures which are called as pyrenoids. They are present. The main function of pyrenoids in Spirogyra, pyrenoids has different functions to perform in different organisms. But specifically in Spirogyra, these pyrenoids, they help in fixing carbon dioxide. Okay, so this is the basic structure of a alga Spirogyra. Now, when due to wave action or due to, suppose if this cell become old and there would be another cell here. So if this cell suppose become old and it needs to be removed from the body of Spirogyra. So this will break off, die off, break off, and these two fragments, one here and one here, will get separated and then further grow into new fragments on its own. Okay, so this is called as fragmentation. So this fragments will get separated from this fragment. Okay, because of the death and decay of this old cell. So here this process is called as fragmentation. Understood? Is it clear till here, bache?
Yes,
Are you through with the two methods? One is fission, another one is fragmentation. Is it clear to all of you? Okay. Then revise and come next week. Right? Okay. That's all for now. I've taken 10 minutes extra of your class. It doesn't matter. I want to finish it. I wanted to finish it. Okay, bache. Thank you.